We chose to do our current events project on a paper by Lawrence et al., which investigates the effect of temperature on the intrinsic flexibility of DNA and its interaction with architectural proteins. Let's start by talking about the structure of DNA. DNA is classified as a semi-flexible polymer. What this means is that at a local level, that is when we're talking about smaller than 150 base pairs or shorter than 50 nanometers, DNA is rigid and not bendable. However, in a scale bigger than this, DNA starts becoming more flexible and automatically sorts into random coils. While DNA has an intrinsic ability to bend, there are specialized proteins that ease this process. Such proteins are called architectural proteins. Few of them are listed below. Histones, which is found mainly in eukaryotes and wraps the DNA into nucleosomes. HU proteins, the main bending protein in E. coli, and then Sul7 and Crane7, which are structurally similar and are found in archaeal organisms. The experiment done by the researchers aims to investigate how the flexibility of DNA changes between the temperatures of 23 to 52 degrees centigrade. The authors analyzed this relationship by tracking the change in persistent length. Persistent length, an often used technique in biochemistry, is the quantitative measure of intrinsic rigidity of any polymer. Let's talk about the experimental procedure. They started the experiment by purifying the proteins Crane7, Sul7, and HU from E. coli. The genes encoding for each of these proteins were obtained using a plasmid and then were chemically forced to express in a culture. Then they moved on to isolate DNA strands. They obtained different DNA substrate, each 685 base pair longs, using the technique of PCR. The substrate were N-labeled and then varied in their GC content. The movement of this DNA substrate was then tracked using the tethered particle motion technique. It's a widely used technique to analyze the motion of a polymer in an aqueous environment. One end of the DNA was attached to a metal bead, while the other was attached to a rigid surface. The movement of DNA then could be easily tracked by recording the movement of attached beads. The X and Y movement coordinates were recorded and used to calculate the root mean square, or the RMS value, of excursion of each DNA. This RMS value was later used to calculate the persistent length. The temperature for this experiment was controlled using a custom-built temperature system. The following video shows the movement of tethered particle attached to a polymer, which was used in this experiment. There are three important findings of this experiment. First of all, the author concluded that increasing temperature enhances DNA flexibility by significantly decreasing the persistent length. In the graph on the right, you can see the curve used to convert RMS values to persistent length. It also consists of a computer-built model of DNA at 23 degrees Celsius, shown in black, and 52 degrees Celsius, shown in red. As you can see, at 52 centigrade, DNA is able to bend more due to its increased flexibility, thus making it more compact. The graph on the left shows the values for RMS and persistent length at different temperatures. As you can see, the persistent length decreases with increasing temperature. One can also observe that there is significant difference in persistent length between DNAs of varying GC content. This indicates that while architectural proteins do help in bending, it is an essentially intrinsic quality of DNA. The next set of findings is in regard to the architectural proteins Crane7 and Sul7. They found that increased flexibility at higher temperatures led to greater affinity between DNA and these proteins. However, compaction of DNA at higher temperatures was independent of these proteins. Thus, as DNA gets more flexible, it seems to not require the architectural proteins Crane7 and Sul7 as much. The case in HU proteins, however, was very different. Not only binding affinity between DNA and HU proteins increased at higher temperatures, it also led to greater compaction. For the same HU concentration, DNA persistent length changed significantly between 23 and 37 degrees Celsius, suggesting major role of HU protein in DNA compaction at higher temperatures. Therefore, in conclusion, higher temperatures seem to increase the binding affinities of architectural proteins, but this does not, however, lead to greater compaction in all cases. Thus, this particular topic requires further research. There are some big implications of this research. First of all, the authors point out that most of the studies regarding DNA organization and compaction is done at room temperature. However, as the results of this experiment shows, the temperature is a major force in DNA compaction and thus needs to be considered for these types of studies. The research results point out that the temperature becomes especially important when considering the organization of organisms that thrive naturally in temperatures that are different than the room temperature. Moreover, this research also provides some evidence that various organisms use temperature as a secondary gene regulation system. Fluctuation in temperature can lead to compaction or extension of DNA, thus exposing different parts of it and leading to varied gene expression. Now, we would like to leave you with some questions to consider. Thank you for watching.